So we'll just wait for 5 more minutes and then we'll start the class, okay? Okay, so a very warm good evening to everybody. So as we were studying in the last week class about plant developmental physiology and plant developmental biology, so in today's class we'll continue some of the plants question, and after continuing the plants question, we'll move towards the other important aspect of developmental biology, where we'll be understanding the developmental biology phenomena which occurs in mammalian cells or mammals. So, just a minute. Okay. Okay. So, good afternoon, everybody. Good evening, everybody. So, in the last class, as we were studying about the plant developmental biology, in this class, as I just mentioned, that yeah, first we'll do some of the questions which were left for plant, and then we'll switch to developmental biology in mammalian systems in mammals. Okay, so I'll first start with the questions of plants, and then we'll move to the animal, especially the mammalian system. Okay, so the first question is, long day plants LDP were divided into two treatment groups, each group consisting of 8 plants, and were grown under different conditions. Group A was grown under complete darkness. So this is the group A, which was grown under complete darkness, while group B was grown under 13 hours of light and 9 hours of darkness cycle. And the following observations were recorded. Based on these observations, identify the correct inferences. Okay, and the options are: Group B may have a loss of function mutation in TFL1. Group A may have an overexpression of FT. Group B may have a mutation in the LF5 binding domain of FT. Group A may have had an exogenous application of CO in the SAM. Group B may have had a loss of function in IFI. And Group B may have a gain of function mutation in FLC. So I'll give 3 minutes to solve this question. And after 3 minutes, I will switch the screen. So you can solve it. Think of all the options and then think of what is given in the graph. Also, note that in the y-axis, they have mentioned the number of plants that are transitioning, that have transitioned to the flowering status. That means in group A, we can see that there are more number of plants that have transitioned to flowering, whereas in B, there are none which have transitioned to the flowering status. Okay, now I will switch the screen. Hmm. Okay, A, C, and D. Okay, so A, that is group B, may have a loss of function mutation in TFL1. C, that means group may have a mutation in the LFY binding domain. And D. Okay, fine. So let us try to analyze. Let us see what are the correct options first and then we'll analyze. So the correct options are these. Group A may have had an overexpression of FT. Group A may have had an exogenous application of CO in the SAM. Group B may have had a loss of function mutation in the LFY gene. Group B may have a gain of function mutation in the FLC. Just a minute. A, C, D. Okay. Now, let us try to understand this question. Okay. I'll try to do full screen so that you can understand it in a better way. Okay, so the question says that long day plants. So these are long day plants. That means they require more of the sunlight. Yeah, sorry. So long day plants were divided into two treatment groups. So you can see that there are two treatments group A and group B. Each group consisting of 8 plants and were grown under different conditions. Group A was grown under complete darkness. So it was grown under complete darkness, while group B was grown under 13 hours of light and 9 hours of darkness cycle. So यहाँ पे it is 13 hours of light and 9 hours darkness. Based on the following observations, correct, we have to identify the correct statement. So the first statement says that group A group B may have had a loss of function mutation in the terminal flower 1 gene. TFL means terminal flower 1. So is it correct? Is it a correct statement? Yes or no? It's not a correct statement. Why? Because the gene TFL1, it stands for terminal flower 1. This gene functions to delay flowering and maintain the vegetative growth. So the function of this gene is to delay flowering. Okay, this gene works to delay flowering. So a loss of function mutation in TFT1 gene would result in uh, early flowering because the gene represses the floral transition. But in group B, you can see in group B, the plants are exposed to normal light conditions. If group B have a loss of function mutation, uh, yeah, if group B has a loss of function mutation, that means A option should be correct. I don't know why they have not given A option as correct, but according to me, I think A option should be correct because this TFL1 gene, it is there is a loss of function mutation. So when there is a I have written terminal only. I'm sorry for my bad handwriting. I have written. I'll just rewrite it again. Okay, this is terminal only. My writing is, and this gene is responsible for delaying. Okay. Okay, okay. Acha, acha, it is group B. Ha, okay, then it is fine. See, I thought it is group A. Now you see that this gene is responsible for delaying the flowering process. Now, if there is a loss of function mutation in this gene, that means the flowering process should be increased or it should be improved. But you can see that in group B, the flowering process, if there would have been a flowering process, then there should be some number of plants that have transitioned to the flowering status, but you can see that none of the plants transition to the flowering status. That means that group B does not have a loss of function mutation in TFL1 gene, because if there would have been a loss of function mutation in terminal flower 1 gene, then there should be some plants that have transition. But since there are no plants, therefore option 1 becomes incorrect. Then your second option is group A may have had an overexpression of the FT locus. FT is your flowering locus T. Okay, so group A, it is the correct option. Why? Because FT, it promotes flowering. Okay, but the fact that group A is in complete darkness, it means that this has happened because of overexpression of the flowering locus, and this overexpression has subsided the absence of light, and therefore there is increase in the expression of flower. Because this flowering locus, in normal condition, this flowering locus will help uh, in the flowering of the plants in response to light conditions. But if there is an overexpression of this FT locus, that it can kind of override the absence of light, which ultimately leads to flowering even without the normal day night cycle. So because there is an overexpression of FT, which is a flowering locus, therefore option A can be treated as a correct option. Although, always remember that these are all the assumptions. See, here it is written may have an overexpression. So, we are just assuming that the increase in the number of plants that have transitioned to the flowering status could be a result of either some of the following options. Either there could be an overexpression of 50 or some other options that we'll see. Then the next option is group B may have had a mutation in the LFY binding domain of the flowering locus T. So, flowering locus T, it is a key protein that promotes flowering that we have already just discussed, but it also interacts with the LFY domain to trigger the floral transition phenomena. Here it is written that group B may have had a mutation in the LFY binding domain of flowering locus. So, if there is a mutation in the LFY binding domain, that then what would happen? It would disrupt this interaction, which will prevent flowering even under the appropriate conditions. Okay, so could you understand? Could you understand why? Sorry. Huh. So that is why this option becomes incorrect. Then the next option is group A may have had an exogenous application of constants, CO means constants, in the sap. Now this CO, which stands for constants, CO stands, so CO basically is responsible for promoting the flowering. How it promotes the flowering? It promotes flowering by inducing the FT expression in response to light. However, in group A, there is complete darkness. This means that there has been an exogenous application of the CO through the shoot apical meristem, SAM, which has triggered the flowering despite the lack of normal light signal. Could you understand? What I'm trying to say that group A may have had an exogenous application of CO constants in the shoot apical meristem region. Why? Because this CO is responsible for promoting flower in response to sunlight. But since this group A plants were experimentally put in complete darkness, this means that these plants have received an exogenous supply of CO in the shoot apical meristem region, which has triggered their flowering despite the lack of normal light. So this is correct. Then group B may have had a loss of function in the leafy gene. So this is LFY leafy, LEAFY leafy, that is the full form. Now this leafy gene again, it is responsible for maintaining the floral stem identity. This leafy gene is responsible for maintaining the floral stem identity. Okay, so in group B, uh, there are no flowering that has happened. So it indicates that there can be a loss in the function of this leafy gene that has led to no flowering plants. Okay, because this leafy gene is also responsible for flowering in a way that it maintains the floral stem identity. So since there is a loss of function mutation in the leafy gene, so the floral stem identity is not maintained, and therefore there are no flowering plants in the group B. Okay. Then group B may have a gain of function mutation in the FLC gene. What is FLC? It is a flowering locus C. Now, what is the function of FLC gene? This flowering locus, there's no space, so I can write here. Okay. FLC is a flowering locus C, which is important for inhibiting flowering. Okay, this FLC locus is responsible for the inhibition of the flowering. Now, if this is there, that means there won't be any flowering. And since group B has no plants that have transitioned into the flowering status, this means that there might be a possibility that group B may have had a gain of function mutation in FLC gene. So when there is a gain of function mutation in the FLC gene, that means even now there are even more rigorous chances that in group B, no flowering would take place. So this is again the correct option. Okay. Now we'll move to the next question. Next question is, which of the following statements regarding flowering and its pathways are correct? Long day plants grown under 12 hours light but 12 hours dark interrupted with white light induces flowering early. Long day plants which are grown under 10 hours of light but 14 hours dark interrupted with white light induces early flowering. Vernalization is induced under short days by FLCG. Inhibition of SOC1 in vernalization pathway induces flowering. Gibberellic acid or gibberellic acid, whatever you pronounce it as, mediated repression of DELA enables FT expression. Loss of function mutation in the SCP genes inhibits reversal to vegetative fate. So, I will give you 3 minutes. Oh, sorry. Oh, okay. So, I'll give you 3 minutes to solve this question and then we'll switch. 2 more minutes. Hmm. Okay, now we'll switch the screen. So, let us see what you have answered. A, B, and E are correct. A, B, and E. Okay, good. Okay, so let us now try to analyze each and every statement so that our concepts gets clear. So, which of the following statements regarding flowering and its pathways are correct? Okay, so the first option is long day plants grown under 12 hours light but 12 hours dark is interrupted with white light induces early flowering. So, this is a correct statement. Why? Because long day plants they flower when exposed to longer periods of light, that we all know, and interrupting the dark phase with white light it stimulates extended daylight, so it will promote early flowering. See, the dark phase is interrupted with white light. That means what is there? There is extended white light period. We are giving extended light period. So when there is extended light period, that means the flowering would happen early. Then therefore, with the same logic, your B option is also correct. Long day plants grown under 10 hours of light but 14 hours of darkness interrupted with white light induces early flowering. Now you can say that here the light phase is 10 hours but 14 hours is dark phase, and how can early flowering take place? But you need to understand that this 14 hours of dark phase is not a complete dark period. You see, it is interrupted with white light. So there is an extended light period. So therefore, flowering would induce early. Then vernalization is induced under short days by FLCG. Now this is an incorrect statement. Why? Because vernalization is the process by which prolonged cold induces flowering. Okay, prolonged cold, it induces flowering. So it actually works by repressing the flowering locus, that is FLC. So vernalization happens by repressing the flowering locus C. So it is not induced by FLC. So this is just the opposite statement. So it is a wrong statement. Then inhibition of SOC1 in mineralization pathway induces flowering. So this is again incorrect because SOC1 stands for suppressor of overexpression of constans 1. SOC1, I'll just write it here. SOC1 stands suppressor of overexpression of constans 1. So inhibition of SOC1 in mineralization pathway induces flowering. This is again an incorrect statement. I don't know what happened to the settings. Okay. Why? Because SOC1 is a gene that promotes the flowering process. So, inhibition of SOC1 would delay the flowering process. It will not induce the flowering process. Okay. So, this is again wrong. Then, gibberellic acid mediated repression of DELA enables the FT expression. This is a correct statement. Why it is correct? Because gibberellic acid plays a role in flowering by repressing DELA protein. Okay, so DELA proteins are repressed by the gibberellic acid, which helps in flowering. So the repression of DELA, it leads to the expression of the flowering locus T, and therefore flowering is induced. So what happens? Gibberellic acid, they inhibits the DELA proteins, and when the DELA proteins are inhibited, then FT, that is flowering locus T, expression is promoted, and therefore it leads to flowering. Then loss of function mutation in the SCP gene inhibits reversal to vegetative fate. So is it correct or not? So SCP means the Sepaleta gene. Now these SCP genes are very much important for flower development. So if there is a loss of function mutation in the SCP genes, then the proper formation of flower would not take place. Okay. And this is about the SCP gene. But does it lead to the reversal of the vegetative fate? No. See, when SCP gene is mutated or there is a loss of function mutation in SCP genes, then the flower formation can be compromised. But it doesn't mean that the plant will revert to the vegetative fate. No, only the floral organ identity is affected. So therefore it becomes a wrong, because this is like a big statement you are giving that if there is a loss of function mutation, then it will go to the reversal of the vegetative fate. No, only the floral identity would be compromised. It will not reverse to the vegetative fate. Fine. Okay, so now we'll go to the next question. Yeah. Identify the correct statements regarding the ABC model. The genes of the subsequent whorls inhibit the genes of the prior whorls to form organ boundaries. B. For a flower to have only petals, loss of function of class C genes is required. D. Class D gene mutant would be sterile. Then, loss of function mutation in SEP will have similar phenotype to leafy mutant. Loss of function mutation in class B genes will feminize the plants. So I'll give you 3 minutes. Try to solve this question and then we'll discuss. So we'll discuss a little bit detail about the ABC modeling, because ABC model is a important topic uh, for your even for your competitive examination point of view, for your CSA and all. So we'll try to discuss this topic, okay, in a little bit detail. 2 minutes more. One minute left. Okay. B is incorrect, rest all are correct. Hmm. Okay, fine. So, first of all, what is this ABC model of plants? So, we know that in plants, the flowers, they are organized into concentric whorls of sepals, petals, stamen, and carpel. Okay? With each of these floral organ types have a unique role in the reproduction process. Fine? So let us just this was the question. Hmm. Hmm. So what is the function of sepals? Sepal basically protects the flower bud. Okay, so it encloses and it protects the flower bud. Petals Petals are the large and the showy organs which helps to attract the pollinators. Stamens Stamens basically produce the pollen grains that contains the male gamete. And carpel has the female reproductive part, that is the ovules, that will fertilize to produce the seed. So these sepals, petals, stamen, and carpel, so they have their own unique roles. Now, the size, shape, number of each of these organ types can be different in different plants. That we know. The size can be different, the shape can be different. How elaborate these sepal, petal, stamen, carpel are, that can be different. But still, one thing remains the same, and that is the general organization of these four floral organs. So, how these organs are arranged in concentric circles, in concentric whorls, it is kind of consistent across all the flowering plant species. Okay, in all the angiosperms, the pattern or the general organization of four floral organ types remains consistent. And this unique combination of the unique organization of these floral organs is studied under the ABC model of plants. So, now what leads to the production of sepals for at the sepal place, petal at the petal place, stamen at the sepal place, and carpel at the carpel place? How it is judged that uh, sepals will protect and cover the petals, and inside the petals you will have carpel and stamen? It is because of the expression of certain genes, and those genes are known as homeotic genes. Okay, so these homeotic genes, where we have studied earlier, can you tell me where we have studied earlier? These homeotic genes we have studied earlier also. Where? In which model else we have studied these homeotic genes? We have studied in Drosophila development. So in Drosophila development, we have studied that whatever the segmentation has happened because of the pair rule genes, there are gap genes, there are pair rule genes. So whatever the segmentation has happened, the identity to each segment is given by these homeotic genes that we have studied in Drosophila development. Same function or similar function is here also, that these homeotic genes basically give rise to sepals at sepal place, petals at petal place, carpel at carpel place. So these homeotic genes can be A, B, and C. Okay. Now, unlike the homeotic genes which are present in the animals that encode the homeodomain proteins, most plant homeotic ABC genes, they include the members of MATS domain class of transcription factors. Okay, so these homeotic genes encodes MATS. I don't know why my pointer is working like this. Let me just try to. Okay. Hmm. Is it working? No. What has happened? Hmm. So I was telling that these homeotic genes in the case of plants, they encodes MAD domain class of transcription factors. Okay. So it is MAD domain class of transcription factor. So specifically in plants, we talk about the Arabidopsis. So in Arabidopsis, A function is provided by epitella 1 with epitella 2. A function is provided by epitella 1 and epitella 2. I think it's better that I write. Hmm. It's difficult to write like this. Okay, I'm sorry, but you have to bear with the extra whatever it is coming because I don't know, there is some settings problem. Okay. B function is provided by two matchbox genes which are known as epitella 3 and pistella, which is known as PI. I think it's better not to write because it's really horrible. Okay. And C function is provided by agamus, which is known as AG. So always remember there are three A, B, and C genes, and these functions are provided by matchbox genes. So A is provided by epitella 1 and epitella 2. B function is provided by two different matchbox genes, epitella 3 and pistillata, which is known as PI. And C function is provided by the matchbox gene, which is known as agamus. In short, we write as AG. Okay, now let us try to see with this figure. This figure will explain it very easily. What happens? Yeah, parasegment. Yeah, Ajay, you are right. In Drosophila development, it happens. Okay, now you see here. Hmm. In normal condition, sepal is formed when only gene A is expressed. Okay, for the formation of sepals, only A gene is required. For the formation of petals, both A and B genes are required. For the formation of the third role, which is stamen, both B and C are required, whereas carpel is formed only by C alone. Okay, so we have the outside is your sepal, then petals, then stamen, then carpel. So sepal is identified, I mean, sepal is designated by only A. Petals is formed by the expression of both A and B. Stamen is formed by the expression of both B and C, and carpel alone is formed by only C. Is it fine? Now, if there is a mutation in the A gene, so if you see here, uh, okay, let it be. Yeah, so if there is a mutation in the A gene, now another thing to remember is that A and C are equally dominant. So if A is not there, then C will come. If C is not there, then A will come. Okay. So I'm explaining it in detail for the people who might watch it in the recorded session. So if there is a mutation in the A gene, then since A and C are equally dominant, so what will happen? That C will come in place of A. C will come in place of A. So now what will happen? C alone will form carpel. So instead of sepal formation in the first role, your carpel formation will take place. In the second, your A plus B forms petals. But since A is mutated, so C is here. So C will form B plus C. What forms B plus C? That is stamen. And this is the normal stamen carpel. So if A gene is mutated, the phenotype would be carpel, stamen, stamen, carpel. Likewise, if the B gene is mutated, so then here instead of A plus B, only B is there. Here instead of C plus B, only C is there. So this will form sepal because A is alone. The role 2 will also form sepal because A is alone. Role 3 will form carpel because C is alone, and role 4 will form carpel because C is alone. Then if there is a mutation in the C gene, what will happen if there is a mutation in the C gene? So, as I told you, that A and C are equally dominant in nature. So, if C is mutated, then A will come in its place. Now, what will happen? The first two will remain same. Here, because alone A, sepal will form, A plus B, petal will form. Now, here, in spite of B plus C, it will be now A plus B because C is mutated. So, now what forms when A plus B is there? A plus B leads to the formation of petals. So here petals will be formed, and here alone A would be there, so it will form sepal. So what would be the phenotype? Sepal, petal, petal, and sepal. So these three mutations case studies are important. So this whole uh, figure is very important. Okay, so you need to learn and understand the concept that how these A, B, C genes, A plus B, B plus C, and C. This is how it works, how it controls the floral organization of a plant in a way that sepal is outside and carpel is inside. Outermost is your sepal, innermost is your carpel. And how mutation in any of the genes can change the physiology and the floral arrangement of the plant. So I hope this ABC model of flower development is clear. Now we'll try to solve the question. The question says identify the correct statements regarding the ABC model. Okay. The first statement is the genes of the subsequent roles inhibit the genes of the prior roles to form the organ boundaries. So, what do you think? This statement is correct or not? This statement is correct. Why? Because interaction between the genes across the roles, it actually regulates your organ identity and form the development of boundaries. For example, if class C gene inhibit class A gene, so see, one important thing to understand is that A and C are equally dominant, and A inhibits C and C inhibits A. So if A is there, C would be inhibited, C is there, A would be inhibited. Okay. So therefore, the first option is correct because these genes they inhibit in such a way to maintain the organ boundaries as class A inhibits class C and class C inhibits class A. Then next is for flower to have only petals, loss of function of class C gene is required. Now we want only petals. Now let us see here. See, for only petal formation, we need A plus B. A plus B will lead to the formation of petals. Okay. Now, here it says for a flower to have only petals. So, what will happen if the C gene is mutated? See, if C gene is mutated, then Role 2 and Role 3 will form petals, but Role 1 and Role 4 will form sepals. So it would not be the case that whole of the plant would be petals. No, it cannot be the case because A is still there. If you want whole of the plant to be petal, it's not possible because if A is also mutated, then alone B cannot form petal. So this particular condition is not possible at all because if there is a mutation in the C gene, then role 4 which is responsible for carpel that will not form, and role 3 which is responsible for the formation of stamen that will also not form because these two roles will form petal, but this will form sepal only and this will also form sepal. So therefore this option becomes incorrect. Then second option is incorrect. Then third option is class D gene mutant will be sterile. Now, you can say that we haven't studied anything about class D genes. We have only studied about A, B, and C. Now, what is D? So, basically, class D genes are involved in ovule development. So, if the genes which are responsible for ovule development are mutant, that obviously means that those plants would be sterile due to defective ovules, which is actually essential for seed formation also. So, therefore, this option becomes correct. Then D is loss of function mutation in Septella will have a similar phenotype to a leafy mutant. So this is correct because both sep and leafy genes they are crucial for floral organ identity. If there is a loss of function in septella, then it will result in floral organs to revert back to the leaf formation. Similar, if the leafy mutants are there, that will also fail to produce proper formation, and therefore the phenotype would can be somewhat similar. So we can say that D option is correct. Then loss of function mutation in class B genes will feminize the plant. Now let us see what is the, from in this figure, what is the male reproductive system here? The male reproductive system is stamen. So if there is a loss of function mutation in the B genes, in second case you see, then sepal formation will take place and carpel formation will take place. That means instead of one female reproductive plant, now there are parts, now there are two reproductive organs, and there is no male reproductive organ. There are two female reproductive organs formed, but there is no female reproductive organ formed. This means that yes, if class B genes are mutated, then it will feminize the plant, because stamen formation can take place only when there is B plus C. If B is mutated, only C is there, then it will form carpel. Okay, so therefore if B genes is mutated, it will feminize the plant because only female reproductive part would be present. Fine. So this is this mutation study, and this particular figure that I have provided here indeed is very important. Okay. Okay. Okay, this is not a plant question. This is also not a plant question. So I think we are done with the plant questions. Yeah, I think we are done with the plant questions. Okay, so now after completing the questions related to the plant developmental biology, now we'll move to mammalian embryonic development. So before moving to mammalian embryonic development, yeah, here, see here also the same things are given in the, I mean in your PDF also same things are there. You can go if, and you can read it. If you haven't read, you can just read and go through it. Just a minute. I don't know whether they have. Huh. So first we'll try to discuss these few points, and then we'll try to solve the questions. Okay. First one is okay, now early mammalian development. So up till now we have studied about the Drosophila development, and we have also studied about the plant developmental biology. Now we'll move to the mammalian developmental biology, which is not that difficult. If you have a basic knowledge of uh, reproduction and female and male reproductive system and how it works, it should not be that difficult, but we'll try to solve questions related to earlier mammalian development as well. So, how your mammalian development differs from your Drosophila development or plant development? Why? First point is that mammalians have very small eggs. Okay, so human zygote is approximately 100 micrometer in diameter, which is less than 1 by 1000 the size of the frog egg. Embryonic development happened inside another organism. Okay, so in most of the cases when we studied earlier, so after the fertilization, development was happening outside, but here the development happened inside the organism, and only after the full developed organism is formed, then it comes out of the body. So after your after the baby is born, only the increase in the size of the already uh, developed organs takes place. What I meant to say is that suppose in plant, your there can be even new branches formations that can take place. New branches are formed, old leaves are weathered, and new leaves are formed, and new leaves are not always formed in the same direction, है ना? They can form in any direction. So that means that there is no control in the formation of new leaves or formation of organ structures, essentially, in the case of plants. But does it happen like this in the case of mammalians? Does it happen that after the baby is born, another finger starts growing? Fifth finger or sixth finger? No. Only the body increases its size and mass. It doesn't develop any new organ after the birth of the baby. So that is what is different. If you cut a finger, the finger will not grow again. So these are few differences that are there in mammalian development as we compare it to Drosophila or plant development. Then very important point is fewer zygotes. That means, for example, frog lay hundreds of eggs. Fish lay many, okay, not fishes, but yeah, just think about very small multicellular organisms. So their formation, I mean, their progeny zygote formation is huge numbers. But in the case of mammalians, zygotes are very few. Then cleavage is so slow. So cleavage is not fast. Then there is compaction and early activation of zygotic gene expression. So these are some of the differences that are there in early mammalian development as compared to plants. One very important difference is that in plants, there is no control in organ formation. Metamorphosis keeps on continuing, but it doesn't happen in the case of mammalians and animals also. So this is the whole female reproductive duct you can see. So in the ovary, your ovulation takes place, and then these fimbriae actually helps in circulating this egg from the duct to through the duct to the uterus or uterine wall, and then this is the infundibulum region where the fertilization takes place. And this zona pellucida, it helps in protecting the egg, and then ultimately, by crossing several stages at morula and at the blastocyst stage, then the implantation takes place. Now you have to understand that when implantation is taking place, it doesn't take place in the middle of the uterus. At the side walls of the uterus, the egg is fixed. The fertilized egg is fixed. So this is just a basic overview of the female reproductive tract. And then the cleavage is asynchronous. That means in many directions the cleavage is taking place. You can see synchronous means that the cleavage plane remains the same. But here you can see the first cleavage plane is this, then the second cleavage plane can be this and this. So it is an asynchronous cleavage that takes place. Okay. So zona pellucida, it prevents your premature implantation, and upon reaching the uterus, embryo digests a whole, possibly by a trypsin-like proteases, which is secreted by the trophoblast in the zona pellucida and comes out. So you can see here, see, once the embryo reaches the uterus, then what happens? Then this zona pellucida is removed and the embryo comes out and it attaches to the endometrium. This is a zona pellucida. With the help of some proteases, it makes a hole in the zona pellucida, and then the embryo comes, it attaches to the uterine wall of the endometrium. And in this process, many kinds of signaling happens, many kinds of uh, cadherins are involved. So cadherins, it helps in adhesion. So you can see many kinds of cadherins are involved in this particular process. Many kinds of pathways such as Wnt signaling pathway also have works over here. So Wnt proteins, they actually induce the trophoblast to secrete a set of proteases that digest the extracellular matrix and enable the embryo to bury itself in the uterine walls. So that means the layer of the extracellular matrix, the, the extracellular matrix layer is digested so that the uh, uh, the embryo can be easily fixed into it. So these are all the basic things. So now the embryo, which is inside the womb of a mother. So, how it obtains the nutrients? It obtains the nutrients from the mother with the help of placenta. So, placenta basically has two parts, chorion and decidua. Chorion is basically the trophoblast, and decidua is coming from the mother, which are the uterine cells. Okay, fine. These are the figures. Now we'll try to solve the questions. Okay, just a basic overview I have given you, because in the last class also I gave a basic overview, because we are where I tried to give a basic overview whenever we are shifting our this thing focus. So now we have shifted our focus from plant to the mammalian system. Okay. So the first question is, which of the following makes it difficult to study the mammalian embryonic development? We have just studied this. Large embryo, fewer zygote, cleavage is faster than amphibians, internal embryonic development. 2 minutes, then I'll switch the screen. One minute left. Okay. Hmm. B and D. Okay. B, that is fewer zygotes, and D, that is internal embryonic development. Yes, it is the correct answer. So here basically we have already just now we have studied that cleavage is slower than that in the case of amphibians. So C options cannot be the correct one. And we have studied that because mammalian embryonic development leads to fewer zygote, and the embryonic development takes place inside the body. So these are the reasons why the mammalian embryonic development becomes a little bit difficult. So yes, next question is, function of Zola pellucida is or are: A, prevention of premature implantation; B, species specific binding of sperm; C, induction of implantation in the uterine wall; and D, digesting the uterine wall. 2 minutes to solve this question. It is right now 7:06. At 7:08, I'll switch the screen. Okay. 1 minute left. Okay. Now I'll switch the screen. Yeah. So A and B you have written. Function of Zola pellucida is prevention of premature implantation, yes, and species specific binding of the sperm, yes. So, uh, this is actually very important that uh, this zona pellucida ensures that sperms from the same species can bind and fertilize the egg. So humans can be fertilized by humans, and this is maintained because of zona pellucida. So a sperm of any other species cannot fertilize humans because of this zona pellucida. So you understand that how much important this zona pellucida is. Then, induction of implantation in the uterine wall, no, because by the time it reaches the uterine wall, zona pellucida is already degraded. Digesting the uterine wall, no. So the correct options are A and B. Yes. Next question is, which of the following structures combines to form the placenta? This also we have just studied: chorion and decidua, zona pellucida and extracellular matrix, extracellular matrix and uterine cells from decidua, trophoblast from chorion, and zola pellucida. 2 minutes. It's 7:09. We'll switch after 2 minutes. So after 7:11. I will switch at 7:12. It's easy one. It's easy only. 1 minute left. Okay. The correct answer is A, which is chorion and decidua. So we have already just discussed that placenta is formed from the interaction between the fetal part and the maternal part, because it has to form a connection between the fetus which is inside the mother. So that means one part has to be from the fetus, other part has to be from the mother. The fetal part is known as chorion and the maternal part is known as decidua. So basically what happens? Trophoblast from the fetal part, that is from the chorion, it invades the decidua, which helps to establish the placenta. Okay. Now zona pellucida and extracellular matrix. No, extracellular matrix, zona pellucida is degraded by the time it reaches the uterine wall. So B cannot be the correct option. Extracellular matrix and uterine cells from the decidua. Now you see here, both are maternal parts. Extracellular matrix is the matrix of the uterine cells, and decidua is also the maternal part. Uterine cells from decidua. So both are maternal. So it cannot be the case, because one has to be from the maternal side, other has to be from the fetus side. Then last is trophoblast from chorion and zona pellucida. No, because zona pellucida is not there itself. So therefore, the correct option is A, which is chorion and decidua. Next question is, the transcription factor responsible for trophoblast formation is A, OCT4, B, NANOG3, C, CDX2, and D, alpha catenin. What is the transcription factor which is responsible for trophoblast formation? 2 minutes. It's 7:40. I will switch at 7:60. One more minute left. Just a minute. Okay. So you have answered it as C, which is CDX2, which is the correct answer. So CDX2 basically plays important role in the differentiation of the trophoblast cell. So here the question is, the transcription factor which is responsible for trophoblast formation is CDX2. Uh, Basically, you have to understand that CDX2, when it helps in differentiating the trophoblast cells, it ultimately helps in the placenta formation, because for the placenta formation, trophoblast cells are important. Whereas OCT4 is basically responsible for maintaining the pluripotency in the embryonic stem cell stage, and other uh, two options, they also are responsible for the stem cell maintenance, but they do not have any role in trophoblast formation. Okay, next question. Hipposignaling pathway is activated in outer cell, endometrium, epithelial, or inner cell mass. 2 minutes. So I'll switch the screen at 7:80. Okay. Okay. Now I'm switching the screen. So the correct what you have answered is D option, which is the inner cell mass, which is the correct answer. So when we talk about the early embryonic development stage, this hyposignaling is important because it regulates the cell fate. So it is active in the inner cell mass to maintain the pluripotency, and it prevents these cells from differentiating into trophoblast cells. So which basically forms the outer layer of the blastocyst. So there are two layers. First is your inner cell mass, and the second one is your blastocyst. Sorry, trophodome. So what layers does trophodome gives? Can you tell me? Trophectoderm is responsible for the formation of which layers? Which layers are formed by trophectoderm? Can you tell me the name of those layers which are formed by trophectoderm? Trophectoderm leads to the formation of three layers, and those layers are known as extra embryonic layers. Embryonic layers are formed by embryonic layers are formed by the inner cell mass, and trophectoderm form the extra embryonic layers. Ectoderm, endoderm, and mesoderm. Okay. So now this hyposignaling, let us just quickly uh, see about this hyposignaling. So why I am uh, focusing on the hippo pathways, because this hippo pathway is important. Okay, so whenever you are studying in development biology, so the study of developmental biology essentially involves the study of some mechanisms and some pathways. Okay, we have studied that various pathways such as your Wnt signaling pathway, AKT pathway, Janus kinase pathway, JAK-STAT pathway. These are some important pathways because these pathways kind of regulate and govern the activity of the various transcription factors in a controlled manner. In the presence of the ligand, something will happen. In the absence of ligand, something will happen. So this is again this those pathways we have studied: epiblast, hypoblast. Yes, Ajay. So those whatever pathways that we have studied in context of Drosophila development, right? The pathways like Janus kinase pathway or Wnt pathway. Those pathways we have studied in context of Drosophila development. So when we talk about mammalian development, hippo pathway is an important pathway. Other pathways are also working in mammalians also. Don't think that JAK-STAT pathway doesn't work in your humans. No, it works in humans as well because it's a general pathway. But specifically, here we're talking about the hippo pathway. What is important over here is that in the presence of the ligand, generally what happens that when the ligand is absent, the transcription factor cannot go inside the nucleus and transcription is ceased. But here what happens that when the ligand is present, then there is no transcription. The transcription happens only when the signaling is off. So it's opposite which is happening. Okay. So this is essentially a human hippopathway that we're talking about. It is based on a kinase signaling cascade, including MST1 and MST2, and as well as LATS1 and LATS2. Here you can see this is MST1 and this is MST. So LATS1 and LATS2. Okay. Don't know why this is not working. Hmm. So see, this is your hmm, this is your inactive hippo pathway and this is your active hippo pathway. Now what happens that when the hippo pathway is inactive, Then the unphosphorylated YAP or TAS, it can enter the nucleus and interact with this tDNA binding protein, which we called as TED1, and target genes are regulated. That means transcription happens. So when the hippo signaling is off, then transcription is happening place. How? Because when the hippo signaling is off, this YAP or TAS, it can go inside the nucleus and it can cause the TED to be expressed, and TED can then start the transcription process. But what happens when the hippo pathway is on? When the hippo pathway is activated, your YAP and hippo on means when the hippo pathway is activated, hmm, then these YAP and as you can see, these are now phosphorylated. And because of this phosphorylation, this YAP and TAS cannot go inside the nucleus and therefore the transcription doesn't take place. So generally, when the signaling is on, transcription takes place. But in this case, it is opposite. When the signaling is off, in that condition, transcription is taking place. And transcription is not happening when the signaling is on. So in this way, this becomes a little unique to study. So you can see here, uh, in the activation of the hippo pathway, in this case, hmm, your MS2, 2, these are the kinases. And this SAV1. So these are your kinase. So they kind of forms a complex. MST1 oblique 2 and SAV1, it forms a complex. And this complex together, it phosphorylates your LATS1 and 2. Now, when LATS1 and 2 is phosphorylated, it uh, basically phosphorylates the downstream molecules, which is your YAP and TAS proteins. Okay. And this phosphorylation asks them for ubiquitination process and then they are degraded. Now in this case you see, when the hippo signaling is off, this complex is unable to phosphorylate the LATS1. Because it is not phosphorylated, it cannot phosphorylate YAP and TAS, and therefore it is not marked for ubiquitination, and therefore it's not degraded and it can go inside the nucleus. So signaling is essentially phosphorylating all the molecules. And when signaling is not there, then the phosphorylation is not taking place. So the signaling is responsible for phosphorylating the kinases. So this is what your hippo signaling is. I hope this hippo signaling is clear. Okay, fine. This was about the hippo signaling. Now we'll move to the next question. We dealt with these questions already. Yeah. Now you see here this question. Female mice were superovulated and allowed to mate. Okay. Fertilization was confirmed with vaginal swab and the female mice were separated into two groups. The treatment group uteri were injected with factor X on every odd day and the embryos were allowed to develop for 11 days, followed by which the uterus was isolated. Okay. Based on the following observations, identify the correct inferences. What are the inferences? What are the options? Factor X may sequester calcium ions. B. Factor X may be an inhibitor of lysyl and prolyl hydroxylase. C. Factor X may be an activator of GSK3K. D. Factor X may bind to endometrium fibronectin and prevent the attachment. E. Factor X may be a protease inhibitor. And F. Factor X may bind to endometrium integrins and prevent the attachment process. So I'll give you 3 minutes to solve this question. It is 7:28 right now. I'll switch the screen at 7:31. Okay? Think of the options, think of the graph, by see the graph carefully and then try to solve the question. Okay. 3 minutes to solve this one. Think of all the options and then try to answer, okay? One minute left. Okay, now I'm switching the screen. Okay. See what you have answered. Yeah, I'm ready. Hmm. Okay. E correct. F. Okay. A, B, C, D, E, F seems all correct. Hmm. Yes. So, indeed, I think all the options are correct here. Yeah, all the options are correct. So, let us see this question. Hmm. Female, what is given in the question is that a female mice was superovulated. Superovulated, what is the meaning of superovulation? So, what is the meaning of superovulation? Can you tell me? What is the meaning of superovulation? What is the meaning of superovulation? Hmm? What is superovulation? Superovulation means the ovulation of more than the required the normal eggs which are ovulated per menstrual cycle. For example, in the case of humans, if we take one egg is ovulated per cycle. But if more number of eggs are ovulated per cycle, then that is known as superovulation. So normally superovulation and all are done during IVF techniques, so that more number of eggs can be extracted from the lady, which can be treated for IVF. So that is the meaning of superovulation. So a female mice was superovulated and allowed to mate. Fertilization was confirmed with vaginal swab, and the male mice were separated into two groups. So after the fertilization, the mice were separated into two groups. The treatment group uteri were injected with factor X. So this is treatment, and this is the control group. The treatment group was injected with factor X on every odd day, and the embryos were allowed to develop for 11 days, followed by which the uteri was isolated. Based on the observations, identify the correct statements. Okay. So, factor first is, it's not working today. Factor X may sequester the calcium ions. See, this factor X was given in the treatment group, which has caused factor X was given in the treatment group, which has caused the repression in the treatment case. So the question option says factor X may sequester the calcium ion. It can be a correct one because calcium ions are critical for cell signaling, particularly in processes which involve cell adhesion and formation of tight junctions. So if there is sequestration means to just sequester means that all the calcium ions are taken up by the factor X. So if all the calcium ions are extracted out because of this factor X, it could impair the signaling which is necessary for the attachment of the embryo to the uterine lining, which leads to reduced implantation. So what we can say that calcium is crucial for adhesion and signaling. If factor X sequester it, implantation could be hindered. So your first option is correct. Then option number B is factor X may be an inhibitor of lysyl and prolyl hydroxylase. Then this is also correct because lysyl and prolyl hydroxylase are the enzymes which which are involved in collagen formation, and it also helps in the remodeling of the extracellular matrix. So inhibiting these enzymes would result in what? There would be poor collagen production, which can potentially weaken the uterine lining and impair the implantation of the embryo. Okay, so collagen is a very important molecule which kind of tightens not only the skin, like we all know that there are nowadays so many beauty products available in the market. They say it has collagen in it, plant-based collagen, meat-based collagen. So what is this collagen actually? This collagen tightens up your skin and gives your skin the important glow and the necessary molecules so that it looks young and fresh. So now, so this collagen not only is responsible for your skin, it is also responsible for the extracellular matrix of the uterine wall. So the remodeling, so what happens, you should understand this thing, that whenever implantation takes place, the extracellular or the epithelial layer of the endometrium, it remodels itself. What is the meaning of remodeling? It turns itself in such a way that it becomes competent enough to take the incoming blastocyst in it. So it has to remodel itself. For example, when guest comes in our house and there are more number of guests, we try to accommodate them. We put chairs, we just shift our whatever things are there little side, so that there is a space to accommodate the incoming guests. Similarly, the endometrium also prepares itself before the implantation, so that the if the embryo is coming, it can properly sit and attach itself. So in that context, epithelial remodeling takes place. And one very important phenomena which occurs during epithelial remodeling is epithelium to mesenchymal transition, which is known as EMT. I don't know if you have heard about this or not. So this epithelial to Mesenchymal transition kind of changes some of the epithelial cells of the uterine wall to mesenchymal cells. So your epithelial cells are round in shape, whereas your mesenchymal cells are somewhat spindle in shape. So some spindle shaped cells are formed during the AMT process. And in the same remodeling process, this lysyl and prolyl hydroxylase, it helps in collagen formation, which helps in the remodeling of the extracellular matrix. So B option also becomes correct. Then C is factor X may be an inhibitor of glycogen synthase kinase 3, which is GSK3. So this is also correct. Why? Because GSK3 is a key enzyme which is involved in various kinds of signaling pathways, especially those which regulate your cell growth, which can regulate your cell adhesion or differentiation. So if there is an overactivation, it could prevent the necessary signaling for embryo attachment. So what happens that if there is overactivation of GSK3, it can disrupt the normal implantation process by affecting cell adhesion and signaling pathways. Therefore, factor X may be an activator of GSK3. Okay, we can say because in factor X has since factor X has overexpressed GSK3K, therefore the implantation has not taken place. So you need to understand the question first. So the question says that whatever treatment has been given to group, this treatment has caused this treatment has affected the implantation process. There is reduced implantation because of this treatment. So now we are trying to see that what this treatment was a factor X which was given for treatment. Factor X was injected into the uterus of the treatment group mouse, female mice. So what this factor 2 could have done, factor X could have done, it could have either sequestered all the calcium ions, it could have either inhibited the lysyl and prolyl hydroxylase. These two are correct. It could have activated GSK3 so that the implantation is inhibited. Then factor X may bind to endometrium fibronectin and prevent attachment. So just now when we were seeing that PDF a little bit, I told you that when endometrium remodeling take place, then several kinds of integrins and fibronectins are activated, which helps in the attachment process. And the expression of these attachment molecules, especially integrin and fibronectin, are very much important because then only the embryo can stick itself to the wall. Otherwise, how would it stuck? So this factor X could have uh, itself bound to the binded to the fibronectin, and now the fibronectin is not available for the incoming blastocyst to bind, and since there is no attachment, then there could be possibly that implantation is inhibited. Then factor X may be a protease inhibitor. Now we have just studied that trophoblast it secretes some kinds of proteases, and these proteases kinds of disrupt the uterine wall also, which helps in establishing a connection between it. So if the protease is inhibited, then again the implantation would be inhibited because the connection cannot be built. So this proteases uh, prevents in remodeling, and therefore the implantation is compromised. The last option is factor X may bind to endometrium integrins and prevent attachment. Just now we talked that integrins and fibronectins they help in attachment, and if there is a binding of factor X with the integrin, then the interaction between the integrins and the blastocyst is blocked. And when this interaction is blocked, then what happens? Blastocyst cannot attach itself, and when it cannot attach itself, implantation would be inhibited. So in this case, all the options are correct. So this question requires what? This question, yes. Ha, you're saying it right. O1 was stimulated multiple X. Yes. So superovulation. Superovulation is also, this is a case of mice, but in humans also superovulation takes place. But most importantly, in the case of bovines, Okay. In the case of bovines, to get higher yielding, to get more calves, higher yielding calves, the higher yielding variety of your uh, buffalo is super ovulated. And when the buffalo is super ovulated, eggs are taken out and then they are transferred to foster mothers. Suppose if a uh, buffalo is a high yielding buffalo, or what is the meaning of high yielding? That means it can produce more milk. So if a more milk producing buffalo is there and we want more than one calves in one cycle, we can we super ovulate so that we can have more number of eggs, and each egg is transferred to a different foster mother. Now that foster mother need not to be, yes, in dairy. So I have worked, so I did my masters from NDRI, which is National Dairy Research Institute. So there they used to regularly do these kinds of experiments. So in fact we went there and we ourselves saw that how, you know, they are injecting the hormones. So basically to cause superovulation you have to inject some hormones. So how they inject the hormones and how they wait, they themselves see the quality of the follicles in the ovary. They themselves, you know, see the touch their ovary by hand. That's a technique that veterinarians do. So uh, it is done. Now when the eggs are taken out and they are transferred to the foster mother, the foster mother need not to be a high yielding variety, because it can be a normal healthy this thing, uh, buffalo. It need not to be a high yielding buffalo because they've already taken egg out. But important thing is, because we are discussing this topic, that the synchronization of the cycle should be there. So when we talk about bovines, in the case of bovines, like in humans, we have menstrual cycle. In the case of bovines, you have estrus cycle. Estrus, estrus cycle. So that, what does it means is that if just randomly I'm saying a number, for example, in the sixth day, you have taken the egg, then the foster mother should also be in its sixth day. Why? So that same phenomena, same mechanism which is happening inside the uh, high yielding buffalo, same, at the same phase your uterus should be in the foster mother also. The endometrium remodeling and everything can be maintained only when the estrus cycle of the foster mother and the donor mother is synchronized, is same. This is very important. So for there are various methods and ways, I'm not going into detail of how you can synchronize the cycle. So you can synchronize the cycle artificially by injecting some things. Okay, so that was just a little bit away from the topic because we are studying about the this thing. That's why I thought of sharing it with you. Anyways, the last question is, which of the following statements regarding mammalian embryonic development are true? A. Loss of function mutation in STAT3 will deplete the stem cell pool. B. Development will be arrested if exogenous CDX2 is injected into in a morula. C. Proper embryo will not form if EOMS is overexpressed within the ICM. Then D. Overexpression of nano will hinder the formation of extraembryonic layer. And the last option, E, OCT4 expression will inhibit the implantation process. It's not a very big question, and I'll give you 2 minutes. It's 7:47 in my clock. 7:49 we'll switch this screen. Okay. Think. 1 more minute left. Okay, now I'm switching the screen. Hmm. So, A, B, C, D, E, all are correct. And I think it's right. One, two, three, four, five. Yes, all are correct. Okay, let us see what are the options. So here we have to select the true statements regarding mammalian embryonic development. The first one is loss of function mutation in STAT3 will deplete the stem cell pool. Okay, so STAT3 is responsible for maintaining the self-renewable property of the stem cells. Self-renewable means that it can renew itself. Stem cells can renew itself. So this self-renewable property is maintained with the help of STAT3. And this STAT3 is specifically important in the ICM region, which is the inner cell mass region. So, yes, if there is a loss of function mutation in the STAT3, that means the stem cell renewable property would be affected. If your stem cell renewable property is affected, that means new stem cells will not be generated. When new stem cells will not be generated, your stem cell pool would get depleted. So, your first option is correct. Second one is development will be arrested if exogenous CDX2 is injected in a morula. Yes, it is correct. Why? Because CDX2 is essential for trophoblast lineage specification. It is responsible for trophoblast lineage specification. So, if we are injecting CDX2 at the morula stage, it can cause misregulation of the cell fate, which can lead to a development arrest kind of a situation. So your B option is also correct. Then your C is embryo proper embryo will not form if eoms is overexpressed within the inner cell mass. Eom means eomesodermin. So basically eoms, which is known as eomesodermin, they are crucial for trophoblast development. Let me write it. I'm not writing today much because this pointer is not working, but chalo, I'll try. Eoms means eomesodermin. Okay. So the option says embryo transfer will not form if eoms is overexpressed within the inner cell mass. True, because eoms is responsible for trophoblast development. If overexpressing overexpression of eoms takes place within the inner cell mass, then the inner cell mass would be pushed towards the trophoblast lineage. So we need trophoblast lineage. Trophoblast lineage is important, but not at the expense of decreasing the inner cell mass. Okay, so trophoblast cannot take the cells and the place of the inner cell mass also in order to grow itself. The inner cell mass is as important as the trophoblast. Both are important. So if there is eoms and eoms are overexpressed in the inner cell mass, then since eomes are responsible for trophoblast development, it will put the inner cell mass also towards the trophoblast lineage. And what will happen? Proper embryo formation will not take place, because embryo will be formed properly only when inner cell mass is also properly formed along with the trophoblast. We need trophoblast, but not at the expense of compromising with the development of the inner cell mass. So your option C would be correct. Then option D is overexpression of NANOG will hinder the formation of extra embryonic layers. So what is the function of NANOG? NANOG is involved in maintaining the pluripotency in the inner cell mass. Okay. And it promotes the formation of a proper embryo. Now it is just the opposite case, just like that of eomes. If eomes are more expressed in the inner cell mass, then inner cell mass will not be formed and only trophoblast will be formed. If nanog is overexpressed, then inner cell mass would be formed, but the extra embryonic layers which are coming from the trophoblast, that would not be formed. Okay, so if there is an overexpression of nanog, this means that the differentiation of the extra embryonic tissue, which is the trophoblast, will not take place. And when the differentiation of trophoblast will not take place, then the formation of extra embryonic layer is also inhibited. Why? Because trophoblast ultimately develops the extra embryonic layers, ectoderm, endoderm, and mesoderm. So from this you can just have an idea that each and every factor is required at a specified time in a specified place. If the and at a specified level, everything has to be maintained in a balanced way. Although nanog and eomes, these are all such an important uh, molecules, such important factors which are responsible for the whole embryo development. But even if these important factors are overly expressed, they can lead to dysfunction or dysregulations, and proper embryo formation can be inhibited. So that means the expression of genes has to be cell dependent, cell specific, time specific, and its quantity or amount specific. It should neither be overexpressed nor be underexpressed. It should neither be expressed before not after. It should neither be expressed at a uh, uh, above place or a below place. It should be expressed just in time at the correct place in correct quantity. Okay. Okay. Last option is OCT4 overexpression will inhibit implantation. Is it true? Certainly it is true. Why? Because OCT4 is a key molecule which is responsible for maintaining the pluripotency. And its overexpression can prevent the proper differentiation which is required for implantation. Specifically, it inhibits the trophoblast differentiation, which is necessary for the embryo to implant into the uterine wall. See, always remember two things. Proliferation and differentiation are two different terms. Never confuse proliferation as differentiation, they are same. No. Proliferation means the cell has to proliferate. Suppose if there is one cell, that one cell will proliferate and form into 10, but the identity of the cell is the same. The same one cell which was there, same will just increase its number. Proliferate means this will increase this number, but this cell is the same. Differentiate means that now this cell will has differentiated means, just a minute. Sorry, I don't know why today this is happening like this. Yeah, it's fine now. Hmm. Let it be. Differentiate means that now this cell has differentiated and it has formed some even enhanced a version of itself. So proliferation may the identity is the same. They're just increasing the number. Whereas differentiation, it has now modified itself. Now it is no more refined. So proliferation and differentiation are two different things. And some enzymes can do both proliferation and differentiation, while others can just do proliferation or just do differentiation. So specifically, when we talk about OCT4 gene, so OCT4 is responsible for the proliferative ability because it maintains the pluripotency. So if it is maintaining proliferative ability, so that means it will not help in differentiation process. So if OCT4 is overly expressed, then what will happen? It will inhibit the differentiation, and the embryo needs to get differentiated now so that it can implant itself into the uterine wall. If it is not differentiated, that means it is not able to release or secrete those molecules which are needed for the attachment or interaction with the uterine wall. Therefore, OCT4 overexpression will inhibit in the implantation. At first go, you can think that OCT4 is a proliferative gene. That how it can inhibit implantation, because it is causing just the proliferation and it is not leading the embryo to get differentiated at all. So from this you can see that OCT4, NANOX, SOX2, these are all the pluripotency maintenance gene. Okay, so I don't know whether we have that or not, but there is a topic which is your induced pluripotent stem cells. Okay, and this in that topic, induced pluripotent stem cells, these genes plays a very important role. OCT4, SOX2, NANOG, and KLF5. These are the genes which are important for inducing the pluripotency in any cell, and it was developed by Yamaha or some scientist named Yamanka, I think Yamanka or something, who discovered this, invented this IPSCs, induced. Yeah, okay. So with this, I think we are done with today's lecture. So today we have solved questions which were related to first we started with plant. After completing plant, we solved the questions which were related to the developmental physiology and developmental biology of the mammals specifically. So today we talked about the mammalian developmental biology and we tried to solve certain questions, and we have also tried to understand the concepts and logic behind it. So if you have any questions, this will be uploaded in the YouTube channel. You can go, you can comment there also, you can ask. So thank you everybody. Okay.